Assalamualaikum and good morning, everyone. Welcome to the very first Biologi Profesor Lectures at Pusat Asasi UiTM Dengkil. My name is Siti Nurfahana and I will be your moderator for today. Now, first of all, I would like to wish welcome back to our guest professor, whom we all familiar with, Yang Bahagian, Profesor Dr. Muhammad Faiz Foong bin Abdullah from the Faculty of Applied Sciences, UiTM Shah Alam. To all biology lecturers who are present today and to all students. Now, before we begin, I would like to invite Muhammad Zaki Nawari from Group S05 to recite the dua. Okay, Zaki?
Bismillahirrahmanirrahim. Allahumma aftah alaina hikmataka wa anshur alaina min khaza'in rahmatika ya arhamar rahimin. Allahumma la sahla illa ma ja'altahu lana sahla wa anta taj'al huzna in syi'ta sahla. Allahumma yassir wa la tu'assir wa tammim bil khair. Rabbana la tuzigh qulubana ba'da idh hadaytana wa hab lana min ladunka rahmah innaka antal wahhab. Rabbana atina fid dunya hasanah wa fil akhirati hasanatan wa qina adzabannar. Wa sallallahu ala sayyidina Muhammad wa ala alihi wa sahbihi wasallam walhamdulillahi rabbil alamin.
Ameen, Rabbi Alameen. Thank you, Zaki, for the dua. Okay, guys, so we all know that Professor Faiz has been with us since last semester, and now he returns to give his lecture on gene technology. And we are very privileged to have Professor with us today, as his main area of research is genetic engineering, and I'm sure Professor can share many of his experience with us. Now, to all students, you can ask, if you have any question, you can ask Professor during our Q&A session, or you can write your questions in the chat box. All right, so I think we can start our Professor lecture now, and without further ado, I would like to invite Professor Faiz to begin his lecture. Okay, Prof.
Okay, thank you, Cik Fana. Okay. Assalamualaikum, salam sejahtera dan selamat pagi. So once again, thank you for inviting me to for this professional lecture. Okay, I just want to verify that you can hear my voice clearly and you can see my presentation. I'll be using a PDF file because PowerPoint seems to be causing trouble.
Okay, we can hear you fine, Prof. Yeah, and your presentation also appear on the screen.
Okay, all right, thank you. And saya juga minta izin untuk recordkan sesi ini. Okay, is that okay with you? All right, okay. Just one moment. All right, okay. We are recording now. Okay, I'll be using the slides that you may have already obtained from your lecturers. Okay, so with a few additions, okay, and some minor edits. Okay, so but in general, the flow will be more or less the same. Okay. So, and if there's anything that looks a bit too much out of the usual syllabus, don't panic. Because I think your lecturer will let you know, okay, which are the parts that will come up in the exam and so on. Okay. All right. Gene technology is something very exciting. Okay? So, in fact, our last 2 decades or so, okay, a lot of scientific advance has been in biology, especially, has been due to the ability for us, okay, to manipulate, okay, things at the genetic level. Okay. So, and in the near future, okay, in fact, a lot of this very advanced technology is already upon us. Is at the front door. Okay. So you may find that almost every aspect of your life, okay, may be governed by gene technology. Okay. You may find that when you want to get married, okay, a computer may choose your spouse for you, okay, no longer your parents. Okay. So by matching DNA, okay, so the computer will decide who's going to be your future wife or husband. Okay. So you may end up getting a handsome Bangladesh here, okay, from halfway around the globe. Okay. Just because your DNA matches very well, okay, and will allow you to produce a offspring with a highly desirable quality. Okay. So it's a kind of a futuristic world that in one way we look forward to. You will solve a lot of problems, okay? But in one way it's a bit scary. Okay, doesn't matter. Okay, so let's take a look at what are the technology that has driven the this gene technology development, okay, for the past few decades. Okay, so I understand that you have been through the first the first 17 or 18 slides, okay, and you have discussed, okay, what are the requirements for you to clone a gene, okay. So the very first gene technology that was developed, I think about 40 or 50 years ago, okay, is the ability to clone a gene, okay. So that allows us to study genes in detail, okay. So genes are very small pieces of DNA. I think you should know that by now, okay, that is there's a copy in each and every one cell in your body, and they produce they carry information, okay, for producing protein, okay. Those protein make up your cells, okay, and run all the biochemical action in your cells, okay, and without them, okay, life could not have been possible, okay. So we have, I think you talk about recombinant DNA technology, okay, and what are the tools that you need to use in recombinant DNA technology. So here I believe that the term recombinant DNA technology is used synonymously as gene cloning. Okay, so firstly you need a target DNA. You need something to clone. Okay, so where do you get the target DNA? Okay, so you could either get it from the cell. Okay, from the organism, or nowadays we use PCR. Okay, but that we'll come to that in a short while. Okay, I'll just go through this slide very quickly as a review. And we have restriction enzyme that allow us to cut DNA very specifically. So this is another major advance in technology. It now allows us to cut DNA in a way that we can control. Okay, all right. So I believe you have gone through this. Okay, how restriction enzyme cut DNA and leave a sticky overhang that we can later use to join back the two pieces of the DNA. So it's a very convenient tool that we have, that nature has provided for us. Okay, and overhang. And once you have cut the DNA, to join them back, okay, you need a gum, all right? And that gum is the enzyme DNA ligase that allows you to join together two different pieces of DNA and form a strong bond, okay, and create a recombinant DNA molecule. Okay, and once you have done that, then once you have those tools, then you need a vector to carry your gene. Okay, and you have talk about bacterial plasmid as a vector. Okay, although there are many kinds of vector. Okay, there are cosmid, bacteriophage vector, yeast artificial chromosome vectors, and so on. In fact, technically nowadays, it's possible to clone a gene into almost any organism: human, mammalian, plants, bacteria, fungi, okay, amoeba, okay, almost anything that you can think of. As long as we can develop a vector for the organism, you can clone a gene into that organism. Although bacteria and yeast are still the mainstream, okay, for cloning. All right, and once you have those two, then you'll learn how to put them together. Okay, that allows you to join two pieces of DNA. Okay, so in this example, it's a bacterial plasmid, which is a round DNA molecule, usually found in bacteria, and the gene that you want to clone. Okay, so by cutting with restriction enzyme, okay, and getting the similar sticky ends, you can join these two together and have a recombinant DNA molecule. Okay, the term recombinant DNA molecule refers to DNA. A mixture of DNA from different sources. Okay. So that's why we call them recombinant. We have combined DNA from two different sources. Maybe one from a human, okay, and the plasmid is from the bacteria. Okay, and then you can join the two different DNA using DNA ligase and finally form your recombinant plasmid. Okay. So once you have the plasmid, then you can introduce it into a host cell. Then the host cell will pick up your plasmid, okay, and replicate the plasmid to many copies. Okay. So that is the basically the very basic of gene cloning. Okay. So again, as mentioned, that it's now possible to use a large variety of host cell. You can clone the bacteria, yeast, mammalian cell, plant cell, and so on. Okay. There's a picture here shows the E. coli, which is still the most popular organism. Okay. All right, and on your plasmid, you should have certain small genes like that, which we call marker genes. Okay, that allows you to select, okay, for the bacteria that has picked up your recombinant plasmid. Okay. Right. I think we will come to all these bits and pieces in a short while when we put them together and look at how we clone a gene. Okay. All right. The last slide that you probably have gone through last before this is this slide on green fluorescent protein, okay, which is a gene that is widely used as a reporter gene, okay, in molecular biology and cellular biology especially. So the discovery of this gene, okay, is actually a cute story. So it was discovered by a Japanese scientist. Okay, so he a Japanese scientist works very hard, and after one night, after a long day at the lab, he decided to go fishing. Okay, and when he sit down under the moonlight, okay, and look at cast his fishing rod, and then he sees that there are a lot of fluorescent jellyfish in the lake that he was in the seaside that he was fishing, and that makes him curious. What makes those jellyfish fluoresce? Okay, so he just take a bucket of water with the jellyfish, okay, and went and go straight back to his lab. Okay, and from there on, he worked and isolated the protein and the gene that allows the jellyfish to fluoresce. Okay. So it's a very small gene, okay, and the good thing is that that gene can be easily cloned and expressed in almost any organism that we have tried so far, and that allows the organism to fluoresce. So you put the gene into a bacteria, the bacteria will fluoresce. Okay. You put the gene into a mouse, you get a green fluorescent mouse. Very cute. Okay, and there was a kind of project some years ago that people are trying to put the gene into the leaf of trees, okay, so that at night, okay, the tree will fluoresce, okay, and you don't need street lamps anymore. Okay, so you have a romantic green color fluorescing leaf that you can bring your girlfriend under and so on. Okay, and this discovery was so important that, okay, it was awarded the Nobel Prize. I can't remember. I think it was in 2006. Okay, so the scientists who discovered the green fluorescent protein, okay, and together with two other scientists, so the Japanese scientists discovered the green fluorescent protein. Another scientist in Texas, okay, further expand on it. So well, everything is green is boring, isn't it? So that guy created, okay, cyan fluorescent protein, okay, yellow fluorescent protein by changing the DNA sequence of the gene. So by mutating the original DNA sequence, okay, so the protein changed from green fluorescent to cyan, blue fluorescent, and yellow fluorescent. Okay, so that was the other development, and that scientist shared the Nobel Prize for this discovery. Okay, and now we have a whole range of fluorescent protein, any color you name it. Okay, yellow, green, blue, red, something called tangerine, okay, and something called cherry, okay, so a whole range of color. So that's what we scientists do, okay. So we look for better solutions, okay, and we improve on the solution, okay. So there's an interesting story on this green fluorescent protein, and they are now widely used as reported genes when you want to study cellular regulation in the cell, okay. Okay, so I think that's where you stop. Okay, and today we'll look at how we are going to clone a gene using a bacterial plasmid. Okay, okay, so sorry, there was a phone call. Okay. All right, okay. So in order to clone a gene, okay, first you need the source of gene. Okay. What gene do you want to clone, and how are you going to get the gene? You can't clone and then something empty. Okay, and in my days, okay, about how old am I? Okay, about 30 years ago. Okay, so the only source that we can put our hands on is the genomic DNA of the organism. Okay. For example, if I want to clone the insulin gene from human, then I have to, okay, look for human cell, okay, extract the DNA, and then from between the DNA, find the gene for insulin. Okay. So once I've found my gene, I can cut it out with a restriction enzyme. Okay, it looks very simple on paper, okay, but there are many hurdles that you will have to plan and overcome. Okay? Because you may find your genes, but then now you have to look for restriction enzyme on both sides of the genes to cut, and that is not easy. Okay, It doesn't matter that we have an easier solution nowadays, and we'll come to it in a short while. Okay, so once you have the gene that you want to clone, okay, and cut with a registration enzyme, then you look for a vector. Okay. The choice of vector depends on the choice of the host cell. Okay. So if you are going to, in the final stage, introduce your recombinant DNA into a bacteria, for example, E. coli, then you want to make sure that you select a vector that can replicate in E. coli. Okay. Otherwise, you will lose your recombinant DNA. Okay. So once you've selected your vector, okay, second step, okay, then you do the same thing. You cut the vector, okay, using restriction enzyme, and in the next step, you join the gene, okay, with your vector, okay, and produce the recombinant DNA molecule. Okay. And in the last step, okay, you transform the recombinant molecule into a host cell, okay, say E. coli, okay, and some E. coli cell will pick up your recombinant DNA, okay, and as the cell multiply, each daughter cell will also replicate the plasmid. And from one copy of recombinant DNA, you end up with millions of copies of recombinant DNA. So that is the essence of gene cloning, from one copy to many copies. Okay. So in those days, we only want to study the gene. So this works well. Okay. But generally now, okay, you not only want to clone the gene, you also want to use the information in the gene to express a protein. Okay. So that will require further additional features on the plasmid. Okay, I don't think it's within our scope, so we will skip that for time being. Okay, so let's take a look at the plasmid. Okay, so so this is an example of plasmid. Okay, so don't worry. Okay, you don't have to memorize this for your exam or whatever. Okay, and this is the way we draw a plasmid. Okay, on the computer. Okay, how we show the information. All right. In reality, they don't look like this. Okay, if you can imagine, they might look something like this. Okay, a double-stranded helix. Okay, that is circular. Okay, and in the cell, normally they are supercoiled. Okay, so you can imagine a rubber band twisted on its own. Okay, and this has been verified. Okay, under the electron microscope, we can actually see the plasmid. Okay, and it looks very much like what we imagine. Okay. So, but on the computer, that's how we will represent the result of this. Okay. All right. Okay. So we have a plasmid here that is called PBR322, one of the very first plasmid to be used for gene cloning. Okay, developed about 40 years ago in the University of Berkeley. Okay. So PBR322 is a small plasmid. Okay, the size is about 4,361 base pair. Okay, so you will compare that to E. coli. The E. coli chromosome is about 5 megabases, 5 million bases. Okay, and this plasmid is only about 4,000 plasmid base pair. All right, and it has 3 genes on it that are important. Okay, so it has a origin of replication. Okay, anybody would like to try why is it important? If you remember the previous 70 slides, why is the origin of replication important? This allows the plasmid to be replicated in the host cell. Okay? Without it, otherwise it'll be lost. Okay? All right, and then it has 2 marker genes. Okay, one marker gene that codes for resistant ampicillin, and the other marker gene codes for resistant to tetracycline, both are antibiotics, meaning that if the bacteria has this plasmid, then it will be able to grow on the media that contain ampicillin or tetracycline. Okay? Because the plasmid allows the bacteria to be resistant to both these antibiotics. Okay? And around it, you have a lot of funny notation. Okay? You can see things like equal RI1, okay? You can see things like Hindi 3, equal RB, and so on. These are the sites where on the DNA where a restriction enzyme will cut. Okay, so and there are many sites there that a restriction enzyme can be used to cut this plasmid. Okay, but again, you have to be careful. Okay? Don't cut somewhere in the genes that are important, right? Okay? Or you will lose the plasmid. Okay, so this is one of the very first plasmid. You can call it the grandfather of all cloning plasmid. Okay, that was developed very early on. Okay, a more modern cloning plasmid will look like this. Okay, it's small again, it's a small plasmid called PAC19. Okay, we try to make them as small as possible, okay, so that it's easier to be accepted by the bacteria and replicated to higher numbers. Okay. So Park 19 was developed in the University of California. Let's explain the UC. Okay, it's smaller than the PBR, about 2,600 base pair. All right, it has amphigenin resistant, the ampicillin resistant gene, which is a marker. It has origin of replication, and it has a LAC-Z reporter gene. Yeah. And interestingly, you look at this part, there's something called the MCS, okay, the multiple cloning site, which contain unique restriction site for a number of enzymes. Okay. So most modern vectors will contain this multiple cloning site, so that you know where to cut. Okay. If you are going to clone a gene, then make sure you use this part, the multiple cloning site. Don't insert it anywhere else, which might disrupt the plasmid. Okay. All right, that could be a bit too technical. Okay, so now we have the plasmid. Okay, and then we are going to find and we have the target gene. Okay, now we are going to cut it with a restriction enzyme. Okay, so generally it's good to use the same restriction enzyme to cut both the plasmid vector and the gene of and your foreign DNA, the gene that you want to clone. Okay, that's probably the pink color region here. Okay, so you use E. coli to class a plasmid vector, then you should also use E. coli to cut your foreign DNA. Although it's not strictly true, but it's probably the best practice, so that you generate the same overhang. Okay, and that allows them to be easily ligated together. Okay, so in the first step, you have cut your target gene that you want to clone and your vector. Okay, and generally we try to cut only once on the vector. Okay, so that's why unique restriction sites are important. Okay. What happens? You cut twice on the vector. You cut once. Okay, you end up with one single molecule. You still end up with one single DNA molecule. Okay, although it's cut now, it's no longer circular. Okay, but you cut twice. Okay, then you end up with two pieces. For example, if I cut here once, okay, and I cut here once, okay, so I end up with two pieces, one big and one small. Okay, if we don't need the small pieces, that's okay. We can lose the small pieces. But if we need the small pieces later, then it'll be difficult to try to find the small piece and ligate it back into your vector. So we always try to use a restriction site that occurs only once on the plasmid. Okay, all right. So, and most cloning vectors, such as bacterial plasmid, will have marker genes. Okay, ampicillin is by far historically the favorite. Okay, because PBR322 and the older generation of plasmid like Park 19 uses ampicillin, so that has been the historical favorite. But you can use any other genes that confer resistance to other antibiotics. Okay? Kanamycin, tetracycline, carbonicillin. Okay, these are all possible marker genes that you can use. Okay, and then the plasmid is combined with the fragment of target gene to form the recombinant DNA, using the sticky overhang that is generated when you cut the DNA. Okay, so if they have the same overhang, okay, and that you will get that you will cut with the same enzyme, then the blue color plasmid and the red color target gene, okay, can easily come together and combine by complementary base pairing. Okay. The sequence of the overhang will be the same, would be complementary, and they can come easy and attach together easily, and that makes it easy for the next step, where we use DNA ligase to seal the gap, okay, and form the bond, okay, strongly, okay, to covalently seal the bond. Okay, so at this step you have your recombinant DNA molecule ready. Okay, the plasmid containing your target gene. In the next step, okay, now that you have produced the recombinant DNA, okay, now we are going to put it into a host cell. Okay, so let's say in this instance we are going to use E. coli, okay, which is the favorite host cell. E. coli, again, for historical reason, okay, we have a lot of information on E. coli. We know how the bacteria behave. We know the physiology well and the genetics well. So, and that makes it an ideal cloning host cell. Okay, because we have so much information and experience with this bacteria, and it's typically harmless. Okay, so there are very little safety issue. Okay. Now the only thing with E. coli is that they don't easily pick up exogenous DNA. Okay, so you try to ask it to pick up a DNA from outside. Okay, E. coli, the usual E. coli, okay, is reluctant to do so. They don't like DNA from external sources. Okay, so to mess up with their regular metabolism. Okay, so you have to do something to the E. coli to induce it into a competent stage. Okay, so and we can do that. We can induce the E. coli cells in the competent stage by treating it with calcium chloride at 4 degrees Celsius in the cold. Okay, so that allows E. coli to pick up your recombinant DNA much more easier. Okay, basically what happened is that the calcium stick to the cell membrane, and because they are positively charged, they attract the DNA molecule that are negatively charged. Okay. So in this step, okay, we allow the DNA molecule to easily attach to the cell membrane. Okay. All right, but that's still just on the outside of the cell membrane. To get the DNA into the cell, okay, we need to do a heat shock. Okay, so while the E. coli is happily swimming in calcium chloride at 4 degrees Celsius, okay, we suddenly raise the temperature to 40 degrees, okay, very quickly, okay, and for a short time, for about 30 seconds. Okay, so what happens when you do that? Okay, the cell membrane actually collapses. Because the cell membrane becomes disorganized, okay, and a lot of holes are formed. Okay, and there is a time when the plasmid will move into the cytoplasm of the E. coli. Okay, so the short time is important because if too long, the cell will die. Okay? If too short, there's not enough time for the plasmid to move in. Okay, so if the plasmid can move into the cell, then you return the cell to the cold, and the cell membrane will reorganize itself. Okay, and you end up with an E. coli cell that has picked up the recombinant plasmid. Okay. Again, it sounds easy on paper, but in reality, very few E. coli cells will pick up the plasmid. Okay, so maybe 10 or hundreds in a million cell will end up picking the plasmid. Okay, and that becomes a transformant. Okay, so the process of introducing the recombinant DNA into an E. coli cell or any bacterial cell is called transformation. Okay, and the resulting cell that has picked up a plasmid is called a transformant. Okay, so you will have a mixture of cells that haven't picked up anything, or cells that pick up the recombinant DNA plasmid. Okay, and some cells will just pick up the empty vector. Okay, so at this stage, okay, at stage 2, when you join the vector with the target DNA, okay, not every vector will pick up a target DNA. Okay, so the overhang, the sticky overhang, makes it convenient for us to join the plasmid with the target gene. Okay, but it also makes it easy for the plasmid to just join back on itself. Okay, so that's also possible. Okay, so at the end of this stage, you actually have 2 products. In fact, many other products. Okay, but the main 2 products will be a recombinant DNA and a recircularized plasmid. Okay, and that can also be picked up by E. coli. Okay, and that's something that you don't want, because you want the E. coli to pick up your recombinant DNA plasmid, the one with the target gene. Okay, right. Okay. Now, once you have the pool of E. coli, and some of them may be a transformant, the last stage is to select for who are the transformant. Okay, which E. coli cell is the one that has your recombinant plasmid, and this is where the marker gene comes into play. Okay, so as we have discussed a moment ago, okay, the marker gene on the plasmid usually confers resistance to the antibiotic. So in this case, it's ampicillin. Okay, so the E. coli that has picked up the recombinant DNA plasmid or the recircularized vector will carry the gene for ampicillin resistance. And when you plate this whole bunch of cell onto a media, for example, neutron agar, that contains the antibiotic, okay, only those cells that has the recombinant plasmid or the vector will grow, okay, and form colonies. Okay. The non-transformant will not grow, okay, and they are eliminated in the process. So these are the one that you have, okay, and we will typically, at this point, we will typically pick a bunch of it, maybe 20 or 30 colonies, okay, and then we screen, we check and see. okay, among these selected colonies, which one of them carries the recombinant plasmid and which one would of them carry only the vector. Okay? And we keep only those that carry the recombinant plasmid. Okay? Which you can further culture into large numbers. Okay, and from there you can extract the DNA, the plasmid DNA, and get many copies of the clone gene, and then you can go ahead and study the genes. Okay. All right. Okay, there's a short video here. Okay, so I'll just play it for you all. Okay, so okay, I can't play video on the PDF. All right. In the case, just a moment, I have to get out of here and share the PowerPoint. All right. Sorry about that. I forgot that there are videos in your slide. Just one moment. Okay. While I quit this. Okay. And bring up a PowerPoint. Okay, here we are. So I do apologize for this. We have to go.
The process of cloning a gene involves insertion of a DNA sequence into a piece of autonomously replicating DNA, such as a plasmid. The transfer of this new plasmid construct into a bacterial cell, and the growth of a population of bacterial cells containing the plasmid. In this way, a clone is generated the exact copy sequence. Plasmid contains bacterial origin of replication and is required for replicating in a bacterial cell. Plasmid also contains a selectable marker gene that makes the bacterial cell transformed with this plasmid, able to grow in specific defined medium.
Prof, I'm so sorry for disturbing, but I don't think we can hear the audio. Hello, Prof.
Oh, sorry.
We can't hear the audio. Yeah, we can't hear the audio. We can see it plays, but then we can't hear the audio of the video.
Sorry about Yes, it's not coming through the microphone then. All right. Do you have access to this video?
Okay.
Are you given a copy of this video?
Well, I
You
don't. Mm.
No. Okay. Then
Maybe.
I'll just play it again. Do you want me to play it again? Okay. Let's
Yes,
try
Prof.
again and see.
Okay.
Okay. How do I get it back to work? Okay. Can you hear it now? Can you hear?
The volume might be a very low, Prof.
Okay. Good.
Mm.
Let me just select the other microphone.
Mhm.
Was that better?
Can
Still can't hear.
maybe Prof can play it again?
Okay, I'll try and see. It automatically off my microphone. Okay. Let me try that. Okay. All right. Reset it. Okay. All right. Let's try again and see.
Okay.
Okay. I'll just make sure that it doesn't off my microphone. All right. All right. Here we go again.
The process of cloning a gene involves insertion of a DNA sequence into a piece of autonomously replicated DNA, such as a plasmid.
Okay, is that okay now?
Okay, Prof.
Okay. All right. Okay. But this is part of your notes, so you might have it in the slide that's given to you by lecturers.
The transfer of this new plasmid construct into a bacterial cell, and the growth of a population of bacterial cells containing the plasmid. In this way, a clone is generated consisting of many exact copies of the inserted sequence. A plasmid contains a bacterial origin of replication that is required for it to replicate in a bacterial cell. The plasmid also carries a selectable marker gene that makes the bacterial cell transformed with this plasmid, able to grow in a specific defined medium. In our example here, the plasmid contains a gene that makes the bacteria resistant to the antibiotic ampicillin. In another location in the plasmid is a copy of the LAC-Z gene that will produce the enzyme beta-galactosidase when the plasmid is inside the bacterial cell. The LAC-Z gene in the plasmid has a region that is recognized by specific restriction endonucleases. Each restriction endonuclease that recognizes this region cuts the plasmid only in this location, so it is a unique site for that enzyme. Most restriction enzymes cut the two DNA strands in different places and thus produce regions of unpaired sequence called overhangs or sticky ends. The piece of DNA we wish to clone must now be generated. In our example here, we will clone the beta-globin gene obtained from human chromosomal DNA. If we treat human chromosomal DNA with the same restriction endonuclease used to open the cloning vector plasmid, a set of specific fragments will be generated that vary in size. Provided that there is not a restriction site inside the beta-globin gene, one of the fragments generated will contain the complete beta-globin gene. However, other fragments of human DNA not containing the beta-globin gene will also be generated. When the fragments of human chromosomal DNA are mixed with the plasmids that have been linearized by the restriction endonuclease, the sticky ends of the plasmid DNA and the human DNA fragments can hybridize with each other, forming double helical structures at the overhangs. These structures can be further stabilized using the enzyme DNA ligase. This enzyme ligates DNA strands by covalently connecting breaks that occur in the DNA backbone. This is exactly the situation at the sites where the double-strand overhangs have now hybridized. DNA ligase closes these strand breaks, making a new circular DNA cloning vector molecule with a human DNA fragment ligated into its unique restriction site. While some plasmids will receive the human DNA fragment containing the beta-globin gene, other plasmids will be generated containing other fragments of human DNA that do not contain the beta-globin gene. Still, other plasmids will simply have sticky ends that find each other and no human DNA will be inserted. DNA ligase will then close these plasmids again, regenerating the original cloning vector. Thus, at the end of the ligation reaction, 3 types of cloning vector plasmids will be formed. The population of various plasmids is then transformed into a population of bacterial cells that have been specially treated to make them competent to receive DNA. This generates a population of transformed bacterial cells which contain cloning vector plasmids with or without human DNA inserts. These cells are then plated onto solid growth media containing ampicillin. Only cells that have taken up a plasmid are able to grow and form bacterial colonies. Petri dishes filled with solid growth media plus ampicillin also contain a colorless substance that the LAC-Z gene protein, namely beta-galactosidase, can convert to a blue substance. After overnight growth, those colonies that arise from cells that received a plasmid without an insert will appear blue. This is because the LAC-Z gene in these plasmids is able to produce a functional beta-galactosidase. In contrast, colonies that received a plasmid with a human DNA insert will be unable to synthesize beta-galactosidase because the human DNA fragment would disrupt the connection between the LAC-Z gene and its promoter. These colonies will appear white. White colonies can then be selected and examined for the presence of human beta-globin gene. A colony containing this gene can then be saved and grown for further study of the cloned beta-globin gene.
Okay, all right. So that's a small story of cloning a beta-globin gene into a vector. Okay, all right. Okay, so what I set up the application. Is there any question at this point? If there's any question that I can help you with, you can ask in the chat box or just open your mic. Okay. All right. If not, then you can just bear with me for a few more minutes. We'll complete this section and then we'll have a short break.
All right.
Okay. Okay. So that was the video that show you how the gene can be cloned into a plasmid vector. At the end of it, there's a mention of a blue-white screening using the LAC-Z beta-galactosidase enzyme. Okay, so you can find the example in the part 19 plasmid. Okay, so this is the part that they're referring to. Okay, so the LAC-Z will allow you to convert a colorless substrate that you can add to your medium into a blue color. Okay, so if you see a blue color, yeah, it means that your E. coli that forms the blue color colony has picked up a vector only, because the LAC-Z is still there. Okay, if the E. coli has picked up your recombinant DNA, the target gene will have been cloned into a part of the LAC-Z. Okay, so you can imagine you cut the LAC-Z into 2 at this point at the multiple cloning site. Okay, and you completely destroy the LAC-Z sequence. Okay, so without the LAC-Z, okay, so the E. coli will not turn blue color. It will turn white color. It will remain white color. And you know that okay, this E. coli has the plasmid because it can grow on ampicillin, but it doesn't have the LAC-Z because it's white color, meaning that the LAC-Z has been interrupted by a clone gene sequence. Okay, so that's how you can differentiate between the E. coli that carries a recombinant DNA plasmid with the gene that you want, okay, or whether it just contains only the recircularized vector. Okay. So as on paper, it looks easy, but when you really sit down to do the cloning exercise, okay, there's a lot of all these small disturbance that might come your way, and you have to solve it. Okay. So we're talking about using a bacteria to clone a gene, make copies of it for study. Okay. If you want to produce a protein from the gene, okay, so it requires a bit more features on the plasmid. Okay, but again, we won't go into that. But this is a good example on how we can use gene technology for the production of insulin. Okay, do you know what's insulin?
Yes.
Okay, what is insulin?
Hormone.
It's a hormone. What does it do?
Turn glucose into glycogen.
Okay, so it controls the level of glucose in your blood. If you have a heavy meal, the glucose in your blood jumps up. Okay, so insulins come into play, and it accelerate the conversion process of glucose into glycogen. So that remove the glucose from your blood. Okay. High blood glucose is not a good thing to happen. You end up with diabetes. Okay. Now certain people are born with a defective insulin gene. Okay, so they cannot produce insulin. So these people cannot control the blood glucose level in the body, and because of that, they develop a syndrome called diabetes, type 1 diabetes, when you're born with it. Type 2 diabetes is a kind of diabetes that you develop because of lifestyle. You eat too much sugar, and your body becomes insensitive to insulin. Okay. Either way, when you have diabetes, and your body cannot produce insulin, then you need to be injected with insulin from an external source. Okay. It's a bit sad because you'll be dependent on external insulin for the rest of your life. Okay. Although again, gene technology offers us a different way out, but that's still far from reality. So before we have gene technology, okay, insulin that's required by the patients are extracted from cows. Okay. So when the cows, so they have to use the cow, okay, take out the spleen and extract insulin from there. So that causes a lot of problem, okay, because one thing, the supply is very limited, so it becomes very costly. Okay. And secondly, because it's a cow protein, it's not a human protein. Okay, although it's very similar, sometimes it can cause an allergic reaction in the patient. Okay, and of course for us Muslim, okay, there is the issue of halal haram, okay, whether the cow is properly slaughtered or not. Although in case of darurat, okay, we are permitted to use insulin from a non-slaughtered cow and so on. Okay, so gene technology allows us to solve the problem by allowing us to produce the insulin in the bacteria. Okay. So we can now take the gene from the human. We know that we have insulin gene because we can produce insulin. Okay, so you can take the insulin gene from the human, clone it into a plasmid, okay, and then introduce it into E. coli, transform into E. coli, and get E. coli to produce the insulin protein. Okay, so in this way now, okay, since the gene is from human, so the protein will be very similar to the human protein, okay, and you do not have a allergic reaction problem. Okay, and large amount of insulin can be produced because now we are using E. coli. We don't have to wait for the cow to die and so on. Okay, and we can produce it in a short time. Okay, so this is one good example where gene technology can be used to solve a major medical problem. Okay, and you may not realize it, but we actually have one of the biggest insulin producer in Asia, in Malaysia. Okay, Biocon Industry, an Indian company. Okay. In a joint venture with the Iskandaria region in Johor Bahru. Okay, so they have set up a very big and very modern facility there to produce insulin using a yeast. Okay, Pica pastoris. This is a yeast that you find on fruits. Okay, they are natural. Okay, so in order to do that, you need an expression vector. Okay, so we have been discussing cloning vector. Okay, but if you want to clone a gene and also express a protein, then you need to have a vector that's a lot more sophisticated. So you can see that on this vector, PPIC-Z, there are a lot more other elements. Okay, we won't go into details. Okay, but you will require things like a promoter to express the gene and so on. Okay, so this is Biocon in Johor Bahru. Okay, if you have time, you may want to persuade your lecturer to organize a field trip down there and look at the current gene technology being used to solve a problem. Okay, So again, just a few more slides. I will go through that very quickly. Okay, so this is traditional gene cloning that we have been discussing about. Okay, so you find you look for a gene, clone it into a vector, and introduce it into a host cell. Okay, the most difficult part is actually to look for the gene. Okay, because the human DNA is about 3 gigabases, 3 trillion bases. Okay, to look for a small gene is a big effort. Okay. Zaman saya, this is during my era. If you can clone a gene, you can be really, really proud of it. You can wear a T-shirt and walk around campus saying that I've cloned a gene and so on, and people will go wow at you. Okay, so fortunately nowadays, okay, things has been made easy by the development of the PCR process, the polymerase chain reaction process. We look at this in a short while. Okay, but basically PCR allows you to amplify any genetic region, any gene sequences, okay, from one copy into many, many copies. So that is the power of PCR. Again, it's another major breakthrough in gene technology. And again, it won a Nobel Prize for the developer. Okay, so by using PCR, okay, you can target the gene region that you want to clone and amplify this particular region and have many copies of it in the test tube. Okay? In fact, PCR itself, the process of PCR itself is almost similar to cloning. Okay? You end up making millions of copies of the same gene sequence. Okay, but this gave us an easy way to get our hands on the gene that we want to clone. Okay, so one way is to extract the DNA, cut it out, and then clone the vector. The other way is to simply PCR it out. Okay, so we'll take a look at it in the next session of the lecture. Okay, so instead of going to cloning a gene by cutting it out, now you can PCR the gene and using the same step, clone it into a vector and introduce it into a bacteria. Okay, it makes things much more faster and much more easier. Okay, certainly a less lot tears. Okay, all right, and I'll just introduce very quickly some recent development in gene cloning. You see a slide with a yellow blue star. It means that this is something that is probably not required in the syllabus. Okay, so just pick it up for general knowledge. Okay, you don't have to memorize the whole thing here. Okay, so TA cloning was introduced about 20 years ago. Okay, to make it easy for us to clone gene. So here we do not require any restriction enzyme, and we do not require DNA ligase. Okay, so we use DNA topoisomerase. Okay, the only requirement is that the fragment that you want to clone must have one tiny extra A overhang, okay, and extra nucleotide on both sides, okay, and adenine, okay, so that, okay, and DNA topoisomerase will grab onto the A and bring it into the vector. Okay, so it works much more faster and it's much more specific. Okay, so one major step in improving the cloning process. Okay, and then sometime about 15 years ago, okay, we developed some scientists developed what is called gateway cloning, and this again doesn't require a restriction enzyme, doesn't require a DNA ligase. It uses a bacterial phage recombinase enzyme to very quickly swap gene fragments. Okay, so it can easily transfer a clone gene from one vector into another vector. Okay, so again another innovation in cloning. And this is my favorite. Okay, this is what a lot of people do today. Okay, you can clone by email. Okay, so this is the dawn of the synthetic biology era. Okay, so if you have the DNA sequence, okay, you can actually synthesize the gene, the DNA using a machine, an oligonucleotide synthesizer. Okay, and then assemble them so you can synthesize them in small fragments, assemble them into the gene that you want, and clone it into a vector. Okay, and the best thing is you don't have to do it yourself. As long as you're willing to pay money, some companies will do it for you. Okay, so very simple. Just sit in front of your computer, your email, or even just on your mobile with an app. Okay, send your gene sequence that you want. Okay, provide the vector that you want it to be cloned in. Okay, and pay from your credit card. And in 2 weeks' time, you will receive your Lazada will send your clone gene in a small test tube. Okay, right to your front door. Okay, this is your era. Okay, everything is by Lazada or Shopee. All right. Okay, I think we'll stop for a break now. Okay, about let's say we take about a 15-minute break. Okay, so if any question, you can type in the chat box. Okay, so and then we'll come back here. We'll rejoin back at 10 minutes past 11. Would that be all right? Okay.
Okay, Prof. Sure.
Okay. Thank you. All right. Thank you. I'll see you again then. Okay.
All right. Okay, Prof. Thank you.
Thank you. Okay. Okay.
But we're still waiting for the admin for us to broadcast to the YouTube.
Oh, okay. All right. I'll wait then.
Okay.
Cik Nur dah ada on ya?
Okay. Okay, everyone, welcome back. I hope you guys have a nice break. So let's proceed and continue with our lecture by Professor Faiz. Okay, Prof.
Okay, thank you, Fana. Okay, and thank you for coming back. Okay, so we spent quite a bit of time a moment ago to talk about gene cloning. Okay, and now we are going to move on to other aspect of gene technology. Okay, so whereby together they allow us to do a lot of things. Okay, my face seems to have disappeared. I'm not sure why. One moment. Okay, well, now we are back. Sorry about that. Okay, so the other technology that was developed and allow us to do a lot of things is the ability to clone a gene library, okay, or a DNA library. Okay, so this is a collection of clone DNA. Okay, and they can be constructed in two versions. Okay, where is one where you clone a genomic library. Okay, so that is where you use DNA fragments representing the entire genome of an organism. Okay, so your purpose here is that you want to have a library of clone, okay, that carries every single piece of the DNA in a certain organism. So we call that a genomic library. Okay, so the other version is a cDNA library or complementary DNA library. Okay, and the cDNA library will only contain, okay, coding region of DNA, okay, fragments from synthesized from the RNA population. Okay, so the difference between the genomic library and the cDNA library is that the cDNA library will contain only gene sequences that codes for protein. Okay, whereas the genomic library will contain every single piece of DNA that is in the chromosome or the genome of the organism. Okay, just checking, is the audio clear?
Yeah.
Okay, all right, thank you. Okay, okay. Now, why do we want to do that? Okay, because if you look at the human genome, okay, so we have about 3 gigabases of DNA. Okay, but less than 10% of that actually codes for protein. So a large chunk of our DNA doesn't seem to be doing anything. Okay. In fact, once upon a time, we call them junk DNA. Okay. They consist a lot of repetitive fragments, a lot of vestiges of viral DNA and so on. Okay, but now we are beginning to believe that, okay, within this 90% of DNA in our cells that doesn't seem to be coding for any protein, okay, they do play important role in regulation of gene expression. Okay, they themselves do not code for protein, but they regulate how our genes are expressed. Okay, at what time the gene is expressed and so on. Okay, we are still at a very, very early stage of understanding all this. Okay, but if the cell kept all the DNA, okay, then there must be important. There must be some function with this DNA. Okay, all right, and that is the same with most eukaryotic DNA. So you have you have a lot of introns and you have a lot of non-coding sequences. Okay. Okay. So we'll just take a quick look at the genomic library, okay, which is basically a collection, okay, of genetic information of a certain organism. Okay, and you can use it later on. You want to study the genome of an organism by either sequencing the DNA or cloning different parts of the DNA. How do we make a genomic DNA library? Okay, so basically it's similar to cloning, okay, except that we do it in a really big way. So in the cloning example that we discussed a moment ago, we only wanted to clone one gene, one piece of DNA. Okay, here we don't care. We want the whole genome. Okay, so you extract the entire genomic DNA from an organism. Okay, let's say a human. Okay, and then cut it into small pieces. So all the DNA from a human cell, cut it into a convenient size. Okay, say about 2 to 5 kilobases. Okay, and clone every single piece of this small DNA into a vector. Okay, So can you imagine 3 gigabases of the DNA, 3 trillion base of DNA, okay, and you cut it into small pieces, you probably end up with a few tens of thousands of different clones. Okay, and every single one will be picked up by a vector. Okay, form a recombinant DNA and transform into E. coli. Okay. So you end up with a collection of about, let's say, 10,000, 20,000, 50,000 E. coli clones. Okay, and that is a library. Okay. So theoretically, if you have a large enough library, okay, then every single gene in the genome, okay, will be represented in the library. Somewhere between the 50,000 E. coli clones that you have, okay, will be all the genes that is present in the genome of the human DNA. Okay, it's a big process, and it's a very laborious process. Okay, in order to create a genome DNA library. Okay? I have a friend who used to do it, and every time she do it, she will ask what happened to the whole lab. Okay? Tomorrow, I'm creating a library, so can everybody help to pick colonies? Okay? So and everybody would drop everything and help to pick colonies. So imagine 50,000 E. coli clone. Okay? If there are 10 people in the lab, each people will pick 5,000. Okay? So we will typically start on a Friday afternoon and work until midnight, just to help to pick colonies. Okay? And give a number to all the colonies, okay, and then she will end up keeping 50,000 clones and spend the next 3 years studying the DNA library. Okay, so this is mainly done for research purpose. Okay, you want to know you want to know the type of genes that's in the in the organism and so on. Okay, so the other type of library is a cDNA library, or a complementary DNA library. Okay, so a complementary DNA is a DNA that you make, okay, using an mRNA as a template, okay, messenger RNA as a template. Okay, so here instead of starting with genomic DNA and cutting them into pieces, we start with mRNA. Okay, so to create a cDNA library, okay, we extract the mRNA from a cell. Okay, and then once you have all the mRNA in the cell, okay, we use an enzyme called reverse transcriptase, okay, to make DNA, okay, based on the sequence of the mRNA. Okay, so from the pool of mRNA, you convert them into a pool of complementary cDNA. And again, okay, all these cDNA are cloned into a vector, okay, and you make recombinant DNA plasmid, transform them into E. coli or another cell. Okay, typically it's E. coli. It's still the easiest to work with. Okay, and you keep a stock of again 10,000, 20,000 different clones, depending on the organism that you're studying. Okay. So for a small organism like bacteria, you probably need only about 5,000 clones. Okay. For something as complicated as human, you may need a lot more than that. Okay, so again, this will give you a library of DNA, but the DNA is made from the messenger RNA. Okay. And why do we do that? Because cDNA clones, okay, a common feature is that they do not have introns. Okay? So you will remember from your topic on gene expression. Have you done that? Gene expression, protein synthesis.
Yes.
Okay. Yes. Okay. So you will know that in eukaryotic cell, okay, the genes typically have introns. So a eukaryotic cell will have DNA genes that is a mixture of exon and introns. Okay, so exons carry coding information. Okay? Exons carry information to make protein, but introns do not carry in any information. Okay, and this is expressed as a transcribed as a pre-mRNA, and a second process called RNA splicing. Okay, remove the intron to form the matured mRNA, from which you can translate into a protein. Okay, so the convenient part of using mRNA is that the cell has already removed the introns, okay, and you're left with only the coding sequences. Okay, introns are important for eukaryotic cell, okay, because they regulate how a gene is expressed, okay, and they allow you to join together different exons to produce different variation of a protein. Okay. But if you're going to clone eukaryotic genes directly from the DNA and you have introns, then that can be a problem, okay? Especially if you want to express the gene in the prokaryotic cell. Because prokaryote like E. coli They don't do introns. They cannot handle introns. Okay? They do not know how to process intron, and when they try to express a protein from a eukaryotic gene with introns, okay, most likely they will fail, okay, and you will not get your protein. Okay, so in order to bypass that problem, okay, instead of going for the genomic DNA, we go for mRNA and make cDNA, because we know that in the matured mRNA, introns has been removed. Okay, so we use an enzyme called reverse transcriptase, okay, to synthesize a single strand of cDNA using the mRNA as a template. This is similar to DNA synthesis, where complementary base pairing occurs, okay, but now a DNA is made using an mRNA as a template, okay, and then once you have the first strand of cDNA, okay, the usual DNA polymerase is used to make the second strand, okay, and then you have a complete double-stranded cDNA molecule that you can clone into a plasmid, okay, and transform into a cell. Okay. And cDNA clones, as mentioned, they do not have introns, okay, and that will allow you to use it later on in a prokaryotic host. Okay, so that's basically the process on how to make a cDNA library. Okay, so again, you end up with a big library of clones. Okay, and theoretically, if you have enough clones, okay, then every single gene, okay, in the genome will be represented in your library. Okay, you only have to take the time now to go and find the gene that you want. Okay. The library is already there waiting to be used. Okay. All right, I'm going to skip this the next section to the last, okay, so that you have enough tools to understand how do we apply recombinant DNA technology. So I'll move on first to the next tool that we're going to discuss. Okay, before we discuss application, I'm going to move quickly to the next tool, the gel electrophoresis, okay, which is a technique that allows us to see the DNA. Okay, so we draw nice circles, plasmid, circular plasmid. We draw double-stranded helix DNA, but in actuality, you can't see DNA. Yeah, you can't see plasmid. Okay, so what you see in the lab when you work with DNA, plasmid, restriction enzymes, and so on are just tiny tubes of liquid. Okay, so you can imagine a very small tube, okay, with a tiny bit of liquid, but that contains an enzyme and DNA and so on. Very small amount. Okay, you plan to become a molecular biologist and do research in the gene technology, you have to be be very comfortable with working with very small amount that you can hardly see. Okay, so you can't see DNA. You can probably see them under an electron microscope, okay, but that is something that you cannot do every day. So we can only visualize DNA, okay, by running it on a gel. Okay, so a gel electrophoresis, okay, separate DNA fragment by size or mass. Okay, so that's about all we can do. Okay, to separate. Hello. To separate large DNA from smaller DNA and look at the size. Okay, so simple, but it actually allows us to do a lot of things. Okay, so we start by making a gel. Okay, typically it's made from agarose, okay, or polyacrylamide. Okay, two different types of gel. Okay, polyacrylamide gel are used when you want to have a high resolution, when you want to differentiate between DNA that is of very similar size. Okay, so you want to differentiate a DNA that is a 400 base pair and 420 base pair. You need a polyacrylamide gel because it has higher resolution. Okay. On an agarose gel, they probably run on the same side and you can't see the difference. Okay, so you make a gel, okay, a slab of gel, either from agarose or polyacrylamide, and then you submerge the gel in a buffer that can carry an electric current. Okay. Plain water doesn't work because plain water doesn't carry currents very well, so you need a buffer. Okay, and then you can load your DNA samples onto the gel, and when an electric current is applied, okay, the DNA is negatively charged, okay, because of the phosphate group, okay, and they will migrate towards the positive pole. Okay. All right, so the higher the voltage, the more quickly the gel will run. Okay, but be careful, if the voltage is too high, the gel can melt, okay, and it will cause smearing of your DNA bands. Okay, so in the gel, large fragment will move slower, and smaller fragment will move faster. Okay, and then after that, you can remove the gel, okay, and look at the DNA by staining it with a dye. Okay. You can't see DNA, but you can stain it with certain fluorescent dye such as ethylium bromide. Okay? So when the DNA is combined with ethylium bromide, okay, and you shine a ultraviolet light on it, it will fluoresce a very nice orange color. Okay? You can Google and for some pictures of it. Okay, and right now we have a lot. Ethylium bromide is historically the most convenient dye to use because it's cheap. Okay, but right now we have a whole range of DNA dye that give you a whole range of color. Again, blue, green, yellow, and whatever color that you want. Okay? You can use to track the movement of DNA in the agarose gel. Okay, so this is how it look like. Okay, so you have a gel. You can see this lab of gel on the, and you put it in the tank and cover it with a buffer. Okay, and then you connect a power source. Okay, and you can load your DNA samples on one end of the gel. Okay, and then once they are loaded, okay, then you pass a current through the gel, and the DNA will migrate due to the current. Okay, so the DNA is negative; it will migrate towards the positive pole of your electrophoresis apparatus. Okay. So the larger DNA will move slower, and the smaller DNA will move faster. Okay. The blue color you see on the gel, okay, usually when they show a gel, it's always blue color. It's due to another dye. Okay, we call it a loading dye that you add to the sample so that you can see, okay, where the sample is more or less running. Okay. The blue color will also run with the electric current. Okay, and if you cut your DNA, okay, to different sizes, then on the gel they will run at different rate. Okay, so this is one example of a DNA gel. Okay, so and on top of the wells where you load your sample, okay? And the biggest fragment will be highest on the gel, okay, and the smallest fragment will be at the low end. Okay? So they tend to run faster. Okay, so this allows you to at least, okay, tell the difference between two pieces of DNA, okay, whether they are the same or they are a different DNA, depending on the size of the fragment. Okay, and this is a picture of a gel that is viewed under ultraviolet light. Okay, so when you shine ultraviolet light on it, okay, DNA intercalated with the ethylium bromide will fluoresce a bright orange, and you can see the DNA running on your gel. Okay. Okay. Again, we have a short video here, so I will spend some time. I'll load it up later. Okay, but before we look at the gel, okay, gel electrophoresis. Okay, again, technology has moved on a lot. Okay, so although agarose gel electrophoresis is still very mainstream, we still do it a lot in the lab. Okay, when we want to have a quick idea of what DNA we are working with. Okay, when you do sophisticated methods like DNA sequencing and DNA fingerprinting, okay, we now use capillary electrophoresis. Okay? So because it offers a much higher resolution. Okay? So instead of having a gel on a slab, okay, we have a gel in a very small capillary. Okay, and then you load your DNA. Okay, the same thing happened. Okay, you pass a current through it, and the DNA will move through the gel in the capillary based on size. The smaller size will move faster, and they will move in front. Okay, there's a dye in the gel. Okay, that the DNA will pick up as it move along. Okay, and the light source or laser source will pick up where the DNA is, and the output is generated by computer. Okay? So you don't see a gel anymore. Okay, but you see a graph generated by a computer to show you, okay, what bands you have in your DNA sample. Okay. So the advantage of a capillary electrophoresis is that it can resolve between the DNA fragments that differs by even one base pair. Okay. So two DNA of very similar sizes, maybe 400 base pair and 401 base pair, okay, it can be separated on a capillary electrophoresis. Okay. All right, let's take a quick look at the video. Okay. Again, please bear with me while I set it up again. Oops, where is it? Oops, seems I lost it. All right, here we are. Okay, just a moment. Okay, this one. Okay, I have to make sure that okay, it doesn't off my mic. And here we go.
Prof, I'm sorry, but we can't hear the audio of the video. Prof, if you want, I can play the video for you. If you want to.
Maybe that's one more try. Okay.
Okay. All right. Okay.
Is also reserved for the placement of DNA of known sizes, and then the power supply is turned on. The blue tracking dye is negatively charged and migrates toward the positive electrode, as does the DNA. The DNA backbones contain negatively charged phosphate groups, which are attracted to the positive electrode. The smallest fragments move the fastest, being entangled less in the agarose matrix of the gel. When the blue dye reaches the bottom of the gel, the power is turned off. A fluorescent dye is then used to stain the DNA fragments. A DNA fragment in the size standards lane is the smallest, in this case about 500 base pairs, followed by DNA fragment D. The largest fragment is 23,000 base pairs long. Each band of DNA may contain millions of fragments of the same size. In many cases, a researcher may want to determine which DNA fragment contains a DNA sequence of interest. To do this, the researcher prepares the DNA in the gel to make a copy, known as a blot. First, the gel is soaked in a basic solution so that
Okay, I think we can stop here. The next part is deals with a different technique, which is a lot more complicated. Okay, so that is the basic of running a DNA gel. All right. Okay, any questions so far before we move on? Okay, if there are no questions, then I'll move on to the next one. Okay, all right. Then we will talk about polymerase chain reaction. Okay, so yet another method that was very important, okay, to the development of gene technology and recombinant DNA technology as a whole. So it allows us to do a lot of things that we couldn't have done before. Okay, so the polymerase chain reaction is a method to amplify and make many copies of a specific DNA segment, okay, in vitro, okay, or you can imagine that it's like a replication of DNA in a test tube. Okay, so okay, the tools that are required in the PCR. Okay, so you need a DNA sample. Okay, this is where you will amplify the DNA sequence that you want to amplify. Okay, you need something called PCR primers. Okay, these are short DNA sequences. Okay, and you need a DNA polymerase. Okay, we use something called tag polymerase in this case, and you need free nucleotides before the four nucleotides that make up the DNA molecule, A, T, G, and C. Okay, and then everything happens in a machine called thermocycler. Okay, or amplifying or DNA amplifying machine, or whatever you want to call it. Okay, all right. So the process of PCR is actually a clever adaptation of the DNA replication process that happens naturally in the cell. Okay, if you remember from your previous lecture on DNA synthesis, DNA replication, okay, what happened is that when your cell want to replicate the DNA, okay, it will open up the double-stranded DNA by using a helicase enzyme and a topoisomerase, okay, and then the DNA polymerase can access the nucleotide sequence here and add the complementary nucleotide to form the second strand of DNA. Okay, so we make use of this, okay, in order to amplify a certain gene sequence in the that you are interested in. Okay, so first and foremost, in order to do BCR, you need some information. You can also, you want to amplify a certain gene. Okay, for example, this blue region here. Okay, you need to have some information on the genetic sequences. Okay, so that allows you to design the primers. Okay, so at the very least, you need information, okay, on the two ends of the region. Okay, so that you can design a primer that is complementary to the region. Okay, so for every region that you want to copy or you want to amplify, okay, so you can imagine that the template DNA is a human DNA, and now you want to amplify the beta globin gene. Okay, so you need to know some information. You need to have some information on both end of the beta globin gene, the 5' end and the 3' end. And from there, you design your primer to be complementary to these two ends. Okay, and PCR will amplify everything in between the two primers. Okay, so when you design primers, there are a lot of things to look out for. Okay, so they must be in the correct direction, 5' to 3', and so on. Okay, but that will be a different topic to cover later on some other time. Okay, so right now, okay, so remember that you need two primers, okay, at both end of the region that you want to amplify. Okay, one for each strand of the template. So the primers can be short, about 20 nucleotide base pair. Yeah, that allows you to have enough specificity that they won't stick to other parts of the DNA. Okay. So when you want to amplify a gene, okay, and you design a primer, there's a possibility that your primer might stick to some other similar sequences somewhere else in the genome. Okay, so you have to make sure that it's long enough to be specific, to be highly specific, and it only stick to the part that you want to amplify and not anywhere else in the genome. Okay, so once you have your primers ready to go, the next thing you need is a DNA polymerase to copy the sequence and make the new DNA strand. Okay, so we use a tag polymerase. Okay, so it's a DNA polymerase isolated from the bacteria Thermus aquaticus. That's where the tag came from, TAG. T from Thermus and AG from aquaticus, which live in hot spring. Okay? So this fellow will happily swim around, okay, at temperature of 100 degrees and so on. Okay, so bacteria tend to be very weird. They can live at very high temperature. Okay, so the good thing about the polymerase is that it's very heat stable, okay? And it can function at temperature up to 95 degrees Celsius, okay? Due to the nature that it came from a thermophilic organism. Okay, so in the first step, okay, so the DNA template that you want to amplify is separated, denatured is the word to use. Okay, so the double-stranded DNA is now broken into, not broken, dissociated into two single-stranded DNA. Okay, and then you add your primers, okay, to bind to the site that they are designed to bind to. Okay, and once there's a primer, and you have DNA polymerase, and you have DNTPs, the DNA polymerase will get to work and start making a new DNA strand starting from the primer. Okay? So this is very similar to what happened in your cell. Okay? With a primer and a DNTP and a template, DNA polymerase will synthesize a new strand based on the information in the template. Okay, so this is what happened in one cycle. Okay, so the same thing can happen over and over again. Okay, so in the PCR machine or thermocycler, you can program the machine to change the temperature, okay, at different time. Okay, and the typical thermocycle cycle is we have 3 steps. The first step is denaturation. Okay, so you need to denature the DNA to separate the double helix before the primers can anneal. Okay, So we do that by increasing the temperature to between 94 to 98 degrees Celsius. Okay, so a typical figure would be 95 degrees Celsius. So at that temperature, the DNA will denature. Okay? That's why it's important to use a tight polymerase, because the polymerase is still active at that temperature. Okay, if it's not thermostable, the normal E. coli DNA polymerase will be inactivated, okay, at high temperature. Okay, and nothing will happen in the next step. Okay, so by heating it up to 95 degrees Celsius, okay, you open up the double helix and your single-stranded DNA, and then you drop the temperature to between 50 to 68 degrees Celsius, depending on the sequence on your primers. Okay? If the primers are very specific, then you can go for a higher temperature. Okay, and at this temperature, the primers will come along and bind to your single-stranded DNA. Okay? The binding is specific. Okay, this is the power of PCR. The binding of the primers to the DNA is very specific, okay, so that you only amplify the region that you want to amplify. Okay, so this happened very fast. Okay, in between about 10 to 30 seconds, and then you increase the temperature again, okay, to 72 degrees Celsius, and here is where tight polymerase will get to work. Okay, so there's a primer, there's a template, and we supply DNTPs. So we happily make the second strand of DNA. And from one double-stranded molecule, you end up with two double-stranded DNA molecule of exactly the same sequence. Okay, you can repeat this cycle many times. Okay, so typically we do about 30 cycles. But any further cycle, the tight polymerase will tend to degrade. Okay, and there are too many. If there's too much DNA in the reaction, okay, the reaction will also slow down. Okay. All right. So after two cycles, one cycle from one double-stranded DNA, you have two double-stranded DNA. It doubles. And after three cycles, okay, you will have 4 DNA, and so on. Okay, so the number increased exponentially. Okay, 2, 4, 8, 16, and so on. And at the end of 30 cycles or so, okay, you may have over 1 billion DNA molecule. Okay, that has the same sequence as the original DNA molecule that you want to amplify. Okay? So this is the power of polymerase chain reaction. But okay, from one copy, you can very quickly generate millions of copies. Okay? And the idea is simple. With one copy, you can't do much. You can't even see it or study it. But with 1 million copy, okay, or billion of copies, then you can easily study the molecule. Okay? You can sequence it later on, or you can cut it with a registration enzyme and run it on a gel and so on. Okay. All right, so all this happened in the thermocycle. Okay, now how do we know that the PCR has been successful? Okay, a lot of things can go wrong. Okay, you design the wrong primers, then nothing will happen. Okay, you can run the the PCR product on a gel. Okay, so after doing your PCR in a small tube, you can take a sample from the tube, load it on a gel, and run it and see whether you get the DNA of the expected size. Okay. So again, here is a good thing about PCR. Okay, you know exactly the size of the DNA that will come out, okay, because you design the primers. Okay, so you know how big is a piece of DNA, okay, that you will amplify, okay, and you can see whether you get the same DNA band on an agar gel when you run the result of the PCR reaction. Okay, so in this case, okay, so the first lane is we call it a marker lane. Okay, so you can is a set of standard DNA molecules that we know the size. You can buy this from any supplier, okay, so that we can estimate the size of our DNA band. Okay, so you always have a negative control, okay, and a positive control. Okay, and in both the sample 1 and 2, the PCR was successful, and you managed to amplify the DNA that you want. Okay, so this is classical traditional PCR. Okay? And it's still very important now. Okay? A lot of the experiment in the lab still depend on you being able to successfully amplify a DNA fragment. Okay. Again, things have moved on a lot. Okay, and now we have real-time PCR, which doesn't require you to run a gel. Okay, but this part is a bit laborious. Okay, and annoying. It's not. It's just get a bit boring when you have to run gel every time. Okay, so we now have a real-time PCR, okay, and this is actually the system that is used to diagnose a COVID infection. So when you are suspected of being COVID positive, you go to the clinic. So the nurse will take a swab, okay, and pick up the virus, okay, and then we know the sequence of the virus gene. We can design primers and try to amplify that part of the gene and see. Okay, you have a positive PCR amplification. That means you have the virus. Okay? You don't amplify anything. That means the virus is not there, because the primers are specific. It will not amplify anything else except the sequence that you want to amplify. Okay. All right. So now the SARS virus that causes COVID-19 has an RNA genome. Okay. So virus again they are weird things. Okay. While most other living organisms use DNA as the genetic material, some viruses can use RNA. Okay. And that gives us a bit of problem because PCR amplifies DNA only. It doesn't amplify RNA. Okay. So we have to first convert the RNA into a DNA. Okay. And we have done that before. We can do it here again by using reverse transcriptase. Okay. So as the first step, okay, to transcribe any RNA into a cDNA. Okay. And amplify the DNA using PCR. Okay, so in this case, because then the sample can be infectious, okay, we are dealing with COVID-19 virus, we don't want to run a gel. Okay, so we want to minimize contact with the sample. Okay, so what happened is in that, okay, in the reaction, we also add a special dye. An example is cybro green. Okay, that's why BR green. Okay, so the dye only binds to double-stranded DNA. And when it binds to double-stranded DNA, okay, it causes a fluorescent signal to be produced, and we can measure the fluorescent signal. Okay? So the more DNA, double-stranded DNA that's produced, the higher will be the signal. Okay? You can imagine as you run the PCR reaction and keep producing double-stranded DNA molecules. Okay? Every cycle the number doubles. Okay, so if there's a lot of double-stranded DNA, a lot of the dye will bind to it, and you have a higher level of fluorescent signal due to the binding of the dye with the double-stranded DNA. Okay? And this is how we can see, okay, whether you're carrying a high virus load or a low viral load. Okay, by monitoring. Okay, when does the fluorescent signal begin to happen? Okay, we can quantify, okay, what the amount of virus that you may be carrying in your body. Okay, all right. Okay, so again, there's a video. Okay, I think I'll leave it to I'll let you play it on your own. Okay, okay. From here, I think we have enough information to go back to the application site. You bear with me again. So we'll go back to the application of DNA. Okay. All right. Okay. Of gene technology or recombinant DNA technology. Okay. So as I mentioned in a moment right from the start, so gene technology or recombinant DNA technology revolutionized a lot of things. Okay. It allows us to do a lot of things that we can't do before in much simpler way. Okay, and we will take a look at a few examples. Okay. So in medicine, for example, okay, we can now identify genetic mutation associated with genetic disease, okay, such as, for example, Hutchinson disease, hemophilia, and sickle cell anemia. Okay, so another good example is breast cancer. Okay, so we know that breast cancer is due to mutation in a gene called BRCA, the breast cancer gene 1. Okay, so and it's possible now for you to screen yourself whether you are carrying a mutation that might predispose you to breast cancer. Okay, so the doctor can take a sample of your blood, and then the lab will extract the DNA, and what they will do is they will amplify the BRCA gene, okay, and then the sequence the gene, okay, and compare the sequence to a normal gene, and see whether there are certain and look for certain mutation that may potentially, okay, give you a higher probability of developing breast cancer later in your life. So, and then you have to make a choice. If you do happen to carry one of these, what we call high penetrance mutation, okay, mutation that we know will increase your chance of getting breast cancer, the doctor may actually ask you, okay, do you want to take preemptive action like removing your breast round? Okay, so again, it's a good thing to know, okay, but it may not be a good thing to make decision. Okay, and that's not unique to breast cancer. Okay, there are also similar tests for colon cancer. Okay, so there are certain cancer that we know certain mutations will uh, greatly increase the chance of developing cancer. And if you have that gene, and then do you want to remove your colon? Okay, can we remove the colon? Usus besar, your big intestine, and you still live. Is that possible?
Yes.
It is possible. Just that you can no longer eat Kentucky Fried Chicken. You will be limited to a diet of a liquid food only. So you have to blender everything. I think that's what the Korean celebrity does, isn't it? They blender all the food. They're so busy K-popping, they don't have time to eat, so they blend everything and just drink the liquid. Okay, all right. So there's one way gene technology has contributed to medical side. Okay, and then we're also talking about gene therapy. Okay, so a lot of diseases, okay, a lot of syndromes are due to defective gene that you are born with. For example, diabetes. Yeah, so you have a defective insulin gene. When that happens, okay, it's very difficult to find a cure because you don't have the gene, you can't produce something. You are depending on the external source or whatever that you need for the rest of your life. Okay, so again, DNA technology offers an alternative. Okay, that we may be able to replace the gene. Okay, we can clone a gene into an organism. So if your insulin gene is defective, can we clone a normal insulin gene, a functional insulin gene into you, so that you can produce your own insulin and you no longer have to receive insulin injection every day. Okay, so that's one way that we can do that. Okay, and again, other examples like production of insulin, production of growth hormone. Again, where people who have defects in the growth hormone gene, they don't grow very well. They tend to be stunted. Okay, and production of vaccine. Okay, so so the COVID pandemic, okay, we have seen a lot of different types of vaccine being produced. Okay, and I think the AZ vaccine is an example of a clone DNA vaccine. So we clone the gene for the spike protein of the COVID virus into an adenovirus, a chimpanzee virus that is not harmful, and use that adenovirus to bring the gene into our cell. Okay? And from there, express the protein, okay, the spike protein, and that becomes a vaccine that immunizes you. Okay, so that was the first example. Okay, the technology has been there for a long time, but due to safety concern, it was not pushed very much. But with the COVID being a crisis, okay, they developed the exact vaccine very fast. Okay, so it allows us to produce vaccine very fast now. Okay, so that's medical field, all right. And the second exciting development that was made possible by gene technology is the development of a technique called DNA fingerprinting. Okay, this has been around for a long time actually. Okay, even in my younger days, we have DNA fingerprinting, but that is done using a much more difficult technique, co-Southern blotting. So we won't go into that. We look at the modern version of DNA fingerprinting. Okay. So DNA fingerprinting allows you to do things like forensic criminals analysis. Okay, so if something happened, there's been a murder. Okay, so and you have evidence like blood. Okay, or even just a bunch of hair. Okay, so can we use that to identify the whoever the criminal is? Okay, it also allows you to identify mass disaster victim. For example, if there's been a plane crash, okay, and you want to know, okay, who are the people, which body belongs to who. When you have a plane crash, okay, the condition of the body can be in a very sad state, beyond normal recognition, and you may have to rely on DNA to determine, okay, this is so and so, and this is so and so. Okay, It allows us to study endangered species. Okay, so we don't have to kill the species to identify it. We can just take a tiny bit of DNA, run a DNA fingerprint, and identify which species is it. It allows us to clarify disputed parentage. In fact, this is the main use of DNA fingerprinting. Okay, so if you want to know whether is my parent really my parent, okay, take a blood sample, okay, father, mother, son, and then you compare the DNA fingerprint. Okay, you should have similar DNA fingerprint. Okay, so the father will be different from the mother, but the son should have half the father fingerprint and half of the mother fingerprint. Okay, and then everything is okay. All right. You see something else, then okay, maybe it's time to see a lawyer. Okay, and then it also allows you to identify human cancer. Okay? So certain human cancer has changes in the DNA structure and chromosome. Oops, what did I do? Sorry. Okay, that allows you to do so. Okay, how do we do DNA fingerprinting? So it relies on PCR and agile electrophoresis. Okay, so so in order to do that, you need DNA, sample DNA. Okay, it could be, as I said, blood. Okay, or semen, or even bones. If it's a case that happened a long time ago, and you only have the bones left, even teeth. Okay, hair, saliva, urine, anything that has a tiny bit of DNA. Okay, is there DNA in the hair? What do you think? Rambut ada DNA tak? DNA is found only in live cells.
Ada.
Ada. You watch CSI or what criminal scenes? Okay, most of the time they will extract DNA from the hair. Is that possible?
Yes.
From the hair. Not from the hair, but when you pull somebody's hair, okay, you not only get the hair. At the end of the hair, there'll be a bunch of cells. Okay, so each hair you may get about 10 or 20 cells at the end of the hair, okay? And that is enough for you to extract DNA and do a DNA fingerprint. Okay? So you are attacked, okay, and you can't get away. So make sure you pull a lot of hair. Okay? Hopefully that doesn't happen. Okay, so this is an example. Okay, when somebody enter the house and cause a crime, and then there's blood evidence. Okay, you have suspect. Okay, call them all in. Of course, nobody will admit. All right, and you want to know. Okay, among the suspect, who is the culprit? Okay, you can do a DNA fingerprint. Okay, so take the DNA of the blood from the crime scene, and then take DNA samples from all the suspect. Okay, and then run a DNA fingerprint. Okay, and see who. Okay, which of the suspect DNA fingerprint profile matches the one that you find on the crime scene. Okay, and that will allow you to quickly identify. Okay, the mechanism behind this is that all of us carries a lot of what we call variable number of tandem repeats region in our genome. Okay, these are part of the genome that doesn't code for protein. Okay, so in some coding sequence you do have repeats, but that they are generally outside the coding region. Okay, and they are all over our DNA. We have a lot of this in our DNA. Okay, and each VNTR, okay, can be different between individuals. Okay, for example, you look at these two columns. So this is individual A, individual B. Okay, the same VNTR. Okay, I'm looking at the blue one, okay, on chromosome 16. Okay, individual A on one chromosome has 5 repeats. Individual B also have 5 repeats. But on the other chromosome, remember, human are diploid organism. We have two copies of each chromosome. Okay, so on the other chromosome, okay, individual A has about 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 repeats, while individual B has 2 repeats. Okay, so the number of repeats can be different between individual, and this is what we look for. Okay, we design PCR primers to amplify the entire region. Okay, so the one with many repeats will produce a longer fragment, and the one with 2 repeats will produce a very short fragment. Okay, so when you have enough of these VNTRs, then you can compile a profile. Okay, so now we are using 3 VNTR, red, green, and 16, on chromosomes 7, 2, and 16. Okay, that gives us 6 bands. Okay, and by comparing 6 bands, okay, we can very quickly match, okay, the DNA profile from one individual to another DNA sample. Okay, the DNA fingerprinting that is used by the FBI, okay, I think amplified 13 profiles, 13 different VNTR. Okay, and that give you a unique fingerprint that they say is probably unique, probably with the probability of 1 in a billion. Okay? So the chance of you having the same DNA profile with another individual is 1 in a billion. Okay. Okay, so this is how DNA fingerprinting can be used. So you can use this to check, okay, your parentage, okay, all the way back to your grandfather and so on. Okay, that you should have similar bands to your parents and so on. All right, so what other things that we can do with gene technology? Okay, so we can create transgenic organism. Okay, so we can insert genes, okay, into animals, plants, or almost anything. Okay, as long as the vector is there. Okay, so you can create transgenic mouse, for example. Okay, so for example, the mouse on the left carries a red growth hormone. Okay, and that allows it to grow the bigger size. Okay, so mouse is a popular organism for research. So we tend to use it to study human processes. Okay, but I think recently somebody has done that in salmon. Okay, so and they have inserted a fish growth hormone into salmon, and the salmon grows to grow to about twice the size of a normal salmon. Okay, so that increased production. Okay, and it's good for profit. Okay, at the same time, they also inserted another gene into the salmon that allows the salmon to grow in winter. Okay, so you can see the power of gene technology. Okay, so salmon normally grows in summer. Okay, and it might take 3 or 4 years for you to have a full-size salmon that you can sell for about, let's say, a salmon will be about 300 or 400 ringgit per salmon. Okay, but if you a transgenic salmon that contains an inserted growth hormone gene can grow double the size. Okay, now it's 600 ringgit, and then it can grow in winter. But if you insert another gene that allows it to tolerate cold temperature. Okay, so instead of taking 4 years to grow, now it takes 2 years to grow. Okay, so your profit double. Okay, so that's one example. Okay, and there are people who are also working with sheep and cows. So you can insert a gene into the sheep, okay, that allows it to produce a protein in the milk. For example, you can produce the human growth hormone in the milk. Okay, and later on you don't have to be injected. You can just go there. If the protein has been modified so that it can be absorbed through your stomach, now you can just go there and drink the milk. Okay, no need to inject. Just drink the milk from the sheep or the goat. Okay, so this is a trend we call farming. Okay, you combine the words pharmaceutical and farming. Okay, and it's also being done with plants and so on. In Malaysia, I think RRIM, the Rubber Research Institute of Malaysia, they are trying to express the protein in the latex of the rubber tree. Okay, so that there's a constant production in the latex. Okay, so when you go tapping rubber next time, you don't want the latex. You want the recombinant protein that's produced in the latex. It could be an enzyme. It could be a medicinal protein. Okay, that you can sell at a much higher price. Okay, so these are all the things that we're trying to do. Scientists are trying to do. Okay. All right. Now compared to animals, okay, we have very few examples of genetic modified animals. Okay, because they are difficult to approve. Okay. We have a lot more genetic modified plants that we insert genes in to create new traits or new characteristics. Okay, so we have now corn or jagung that you will insert a gene, okay, from that can allow it to be resistant to drought, so it can grow in dry area. Okay, you have papaya that you can modify the genome so that it becomes resistant to a ring spot virus. Okay, and this particular GM papaya actually saved the papaya industry in Hawaii. they have a very serious problem with the ring spot virus that almost collapsed the entire papaya industry. Okay, but then scientists created a papaya that is resistant to the virus by inserting a gene. Okay, and that allows that papaya to be grown even though the virus is still around. Okay, and we have a lot of soybean that has been genetically transformed. Okay, to resist herbicides and so on. Okay. In fact, most of the corn and soybean that we import now, they are genetically modified. Okay. So if you want non-genetic modified soybean or corn, you have to buy organic soybean or corn. Okay. The jagung manis, the sweet corn, are probably not, okay, because we plant them. Okay. The corn that we buy in bulk to make animal feed, okay, those are most likely genetic modified, and the soybean are genetic modified. Okay. So the next time you drink soybean milk or eat tofu fa, okay, so you may be consuming a genetic modified plant product. Okay. Okay. So this is just a very quick example on how we can use that to improve our agriculture. Okay. So this is one of the very first genetic modified corn, okay, where we inserted a gene from a bacteria, okay, called Bacillus thuringiensis. Okay. So the bacteria produce a protein called a Bt crystal. Okay. Bt for Bacillus thuringiensis. Okay. So we isolated the Bt, the crystal gene from the bacteria, okay, clone it into vector and use the vector to insert the gene into the corn. Okay. So the process is very similar. Cut the gene from the bacteria, okay, like get it into vector, a plasmid, okay, and use the plasmid to carry the gene into the cell of the corn, okay, and insert it into the chromosome of the corn. That is possible in corn, okay. So when that happens, okay, we can grow corn that is transgenic. Transgenic meaning that every single cell in the corn carries a copy of the gene. Okay. So and when the corn produces seed, the seed will also contain the inserted gene, and the next generation will also contain the inserted gene. That's what is meant by transgenic. Okay. So what happened now is that the corn now has a Bt gene from the bacteria, and they can produce an insecticidal protein. They produce a Bt crystal protein, which is toxic to some insect pests. Okay. So when you grow this corn, and the insect pests come along to attack, okay, the insect pests will die when they eat the parts of or the leaf or parts of the corn. Okay. So you have a corn that produces its own pesticide. You don't have to spray. Okay. So you can compare the product from a non-Bt corn. Okay. So you can see the massive damage due to the attack by the pest. But the Bt corn, okay, cantik saja. Okay, they look very nice because the pests, when they try to attack, okay, they will die due to the pesticide. Okay. So this is one example of a transgenic plant, okay, that is being developed a lot in the market. Okay. So besides animal and human and plant, okay, so there are other applications for this technology. Okay, so we can insert a gene into a bacteria, for example, a bacteria called Pseudomonas, okay, that allows it to break down hydrocarbon in oil and clean up oil spillage. Okay. So Pseudomonas itself can break down oil, but not fast enough. Okay. But we can insert other genes from other bacteria, okay, that allows it to do the work much faster. Okay. We can also create microorganisms, for example, the normal yeast, okay, which used to ferment to bake your bread. Okay, you can insert genes that allow them to absorb or detoxify certain metals in the environment. Okay, so you have heavy metal contamination in the soil or in the water. You can just add the organism, and they will quickly bind the metal to themselves, okay, and allow it to be removed from the environment. Okay. So these are all the possible application of gene technology. Okay, all right. There are many more. There are actually many more. Okay, and the application is so wide-ranging that it's quite difficult to cover everything in one single session. Okay, I just have two more slides. You can bear with me. Okay, we'll go straight to the bottom. Okay, what to look out for in the future? So what we have discussed are things that has happened. In fact, some of them are in the past. Okay, so what we are doing now, okay, or in the very near future, are things like that is a lot more sophisticated. So right now we can do next generation DNA sequencing that allows you to sequence a large amount of DNA in one go. Okay, so in the very near future, okay, it may be possible to get to sequence every single individual human. Okay, and by having your genetic blueprint, okay, by knowing the DNA sequence of everybody, okay, there is a lot of possible application. Like we can start predicting, okay, what disease that you may develop and so on. Okay, on hindsight, it's also a bit scary, okay, because you become like an open book. Everybody can read you. Okay, if the insurance company knows that you have a likelihood of developing certain disease, okay, later on, so would that insure you? Okay, will somebody employ you? Okay, so you may become the way we live may change a lot. Okay, but the day is very near, so we can sequence the entire genome for less than 10,000. In fact, less than 1,000 ringgit now. Okay, so 1,000 US dollars, sorry, now. So there might come a day when when your baby is born, okay, doctor dah kata sequence dulu, okay, ambil blood sample and sequence the DNA, okay, and all the data is inserted into a big computer, okay, and everything, and your life is decided by your DNA genome, okay. And then we also have a very, very exciting technology, okay, called gene or genome editing, using a system called CRISPR-Cas9. Okay, so this is a very convenient, very fast and easy way to make changes to DNA sequence. Okay, so previously to make changes to DNA sequences, we have to clone the gene, make the change, and then reinsert it back. But this allows us to make the change directly in the organism without cloning and so on. Okay, also this is getting a lot of attention from the plant people to make genetic modified plants and so on. Okay, and on the horizon is synthetic biology. Okay, so this allows you to clone the gene, not only one gene, but to clone a whole set of genes, okay, to reconstruct an entire pathway in an organism. Okay, and you can control the gene expression using engineering principles. So this is what we have on our front door. Okay, and they are going to impact your generation a lot. Okay, all right. And the last slide, okay, so gene technology is not without controversy. Okay, so there are ethical issues, okay, and that we may have to bear in mind before we simply use them. Okay, so I'll just raise two very quick example. So one is golden rice. Okay, so this is rice or paddy plant where we inserted the gene to produce vitamin A precursor into the rice. The original intention is to solve the problem of childhood blindness due to lack of vitamin A. And this happens in some third world or developing country. So you're lucky in Malaysia, you don't have this problem. Okay, but in some developing countries, about 10% of the children go blind in the childhood simply because they cannot get enough vitamin A. Okay? And one of the way that we thought we can solve the problem is to make a genetic modified rice plant that will produce vitamin A precursor, okay, so that when they eat the rice, okay, they get an extra dose of vitamin A. Okay, but then again, there are a lot of controversy, and until now, okay, we have developed, scientists have developed the rice many years ago. It's been around for about 12 years, but we still cannot plant it, okay, due to ethical. There's still a lot of debate on ethical and safety issue. Okay, so again, okay, later on, one of you may become the minister. Okay, mana tahu kan? Okay, Perdana Menteri kan, and you may have to decide on the issue whether do we want this type of technology? Okay, and the latest one, okay, controversy is when somebody actually went ahead and create the first genome edited human. Okay, so a scientist in China, okay, created the world's first CRISPR twin babies. Okay, what he did was he used a gene editing system to disable a gene called the CCR5. Okay, his intention was good because this gene, okay, is produce a protein that is used by the HIV virus to enter into our cell. When he disabled the CCR5 gene, theoretically the babies will be immune to HIV virus infection. Okay, but we do not know what are the other effects. If we have a gene, it must have some function, and you simply remove the gene. Okay, we do not know what other effect it will have on your body and so on. Okay, again, there'll be a lot of debate on whether this type of research should be allowed or not. Okay. All right. With that, I think I'm on my last slide. Okay. So thank you for listening. And if there's any question, you can either ask on the mic or you can type in the chat box. If I cannot answer today, I'll just pass that answer back to the lecturer some other time. That's possible. Okay. Thank you very much. And if there's any question, I'll be happy to take.
Okay, thank you, Prof. All right, so students, do you have any question for Prof? Okay, Prof. Maybe a bit about the genetically modified organisms. Why do you think people are still being against and skeptical about GMO? Maybe because the GMO would exhibit any kind of harmful effects?
Okay, when a GMO is released into the market, so if it's not released in the market, then we don't have to worry about it. Okay, so a lot of safety assessment and safety tests are conducted, and regulators, for example, in Malaysia, we do have a biosafety law, the biosafety law 2007. If you want to bring in a genetic modified corn, for example, you have to apply for approval. And the regulators will look at the genetic modified plants and decide whether is it safe to be released to the consumer. So that is based on scientific evidence. And in most of the cases, they decide that the risk is very low. But a lot of people are afraid of the technology, because it's like you are doing something that we do not have a history of safety. We do not really know what are the long-term effects. So genetic modified plants has been around for about 10, 20 years. And some people think that there's not enough safety data for us to jump into that. And also people are afraid that when you mess with one part of the genome, so you may have cause unintended effect on another part of the genome. Again, we do not have technology to detect all this unintended effect. So, and probably there's also a segment of the population that simply doesn't like the technology, because they think you are playing with God, manipulating a living organism. So this is the possible reason why there are some objections against GM plants and so on.
Okay, all right. Thank you, Prof. And we hope that hopefully in the future technologies would help. As technologies improve, it could clear up any skeptical feeling that we have regarding GMOs. Okay, thank you, Prof. And students, if you still have any questions, you can still ask Professor if you have any. Or you don't. Okay. All right. So, okay. I think maybe they are, somehow being fascinated already, by Professor's lectures today. All right, Prof. I think that's all the time that we have today. Thank you so much for the clarification and sharing. And we have learned so many things from you. And Prof, before we end our lecture today, maybe you can give our students any advice, because this would be their last semester in Asasi.
Oh, I see. Okay. Just one moment. There's a call coming in.
Okay.
Yes. Sorry. Can you just give me 30 seconds while I just see why somebody is frantically trying to call me?
Okay.
Hello? Hello? Yeah? They are not saying. Okay, sorry about that. Okay. All right. Any advice to give you all? Okay. Well, I presume that most of you will be targeting for a career in medical. Okay, so and all the best in that respect. Okay, so it's a challenging field. Okay, then again, if you happen to, even though after you complete your medical degree, you happen not to like to maybe not to like practice, then you may want to venture into research. Okay, so there's a lot of way you can contribute, okay, by doing research in gene technology for medical purposes. Okay? There's a lot of things to explore there, okay? And they're very exciting technology and opportunities. Okay? And if you decide that you might want to do something else, okay, then again, biotechnology may be one good field to venture into. Okay? So we are seeing a lot of exciting technology coming out on the horizon. Okay, that you can and so on. In any ways, okay. So all the best to your study. This is your last semester. So maintain your humility. Okay. So remember, you can only progress if you have the humbleness to learn and keep learning. Okay. So that is an important, a very important characteristic in life. Okay. I think that's all I have to say to you. You're all bright students. Okay. So tak perlu tegur sangat. Okay. Semua baik-baik belaka kan. Okay.
Yeah, they're all brilliant students.
Yes.
Okay, Prof. Thank you so much for the wisdom words. And I think all of the students would feel very motivated. And we also hope that students would feel very intrigued and also feel very interested with genetic engineering as well. Okay, Prof. Thank you so much for spending your time with us today. And thank you so much again, everyone. So for students, you can find your attendance link in the chat box, and also the QR code will be displayed on the screen shortly. So that's all the time that we have today. Thank you so much for joining our lecture. Stay safe, take care, jaga SOP, and Assalamualaikum and goodbye. Thank you so much.
Okay. That's for you.
Okay, thank you so much, Prof. Come again.
Inshallah.